All right, good morning, everybody. I'm just going to wait another couple minutes here to see if we've got a few more attendees. All right, I think we'll get started here. My name is Liz Murray and I work with Earthmaster Environmental here in Calgary, Alberta. When I saw the note go out from Joe looking for moderators, I said I'd be happy to help out wherever he wanted. And then he stuck me in the Saskatchewan session. So I hummed and hawed about making sure I wore the appropriate colors. So just want to let you know I'm repping the Stampeders today. Welcome to the session. We're going to be hearing from two PhD candidates and a postdoc working in the University of Saskatchewan. First, though, we're going to hear from our sponsor, TerraStrike. So I'm going to let RemTech take over and share a video from them.
TerraStrike is based in New Hampshire. Our manufacturing facility is outside of Toronto in Brampton, Ontario. And we have done work in 5 nations throughout the world. The majority of our work is in North America, Canada, and the US. Been in 23 states and 4 provinces. We've also been in Japan. We've been in the tannery districts of India. And we have done sludge work in Saudi Arabia.
All right, thank you to TerraStrike for sponsoring the session today. This is the session from the University of Saskatchewan. I'm your moderator, Liz Murray, and I work for Earthmaster Environmental here in Calgary. So our first talk today is going to be from Amy Jimmo. She's a PhD candidate at the University of Saskatchewan, and she's going to be talking about the role of carbonates and the effectiveness of biostimulatory solutions for the removal of petroleum hydrocarbon contaminants in cold calcareous soils. So Amy, I'm going to get you to share your webcam and get your presentation going here. And we'll be checking back in at the end. You have 25 minutes. I'll step in and just give you a warning at 25 minutes. And I'm going to let you take it from there.
Great.
I'm going to get you to swap your screen. There you go. Good, good.
All right, so we're showing the right screen. All right, well, thank you everybody for coming to this talk, this early morning presentation. The talk today focuses on the role of carbonates in the effectiveness of biostimulation for remediation of petroleum hydrocarbon impacts in Western Canadian soils. So as we know, when we have a hydrocarbon spill, we're adding additional carbon to the subsurface. Unfortunately, there is not often enough nitrogen and phosphorus in the soil for the microbes to actually be able to use the, sorry, I was filling a few things, but there's not enough nitrogen and phosphorus nutrients in the soil for the microbes to actually use that carbon as an energy source. And that's where biostimulation comes in. Biostimulation is adding a nutrient solution, such as a fertilizer, to stimulate indigenous microflora to use hydrocarbons as an energy source. In situ biostimulation has been around for a long time and is becoming more and more prevalent in northern climates. However, there are still data gaps surrounding the effectiveness of so-called off-the-shelf solutions. This is because a lot of those initial solutions were developed in areas with more homogeneous soils with lower amounts of carbonates. Recent studies conducted on soils from Western Canada have showed a lot of promise. These studies have looked at ways to increase phosphorus availability by looking at sorption, complexation, and precipitation reactions, as well as looking at the addition of a low molecular organic acid or low molecular weight organic acid to chelate calcium and magnesium ions to increase the availability of phosphorus in solution. In practice, addressing a lot of these site-specific factors has worked, but in a few sites, we've seen some stalling. This is believed to be associated with soil buffering capacity and carbonate mineralogy. This is particularly relevant in Canadian soils, Western Canadian soils specifically, because we have high inputs of clay and carbonates from our glaciolacustrine deposits and sedimentary bedrock. These deposits essentially influence the bioavailability of essential nutrients like phosphorus and nitrogen. In order to understand the role that carbonate mineralogy is playing on the effectiveness of our biostimulatory solutions, we really need to understand soil buffering capacity. Soil buffering capacity is the ability of a soil system to resist changes in pH. It's directly related to cation exchange capacity, base saturation, and acid neutralizing capability. So essentially, when you have a high cation exchange capacity, you have a high buffering capacity because there are more exchange sites on the surface of your soil to buffer against the acidic inputs from the biostimulatory solution. In calcareous soils, dissolution of calcium and magnesium carbonates is the main mechanism for buffering capacity. Both magnesium and calcium ions react with phosphate anions, which is the dominant form of phosphorus in the soil. When the acid in the form of our biostimulatory solution is added to these calcareous soils, our carbonates dissolve, allowing for the formation of calcium phosphorus or magnesium phosphorus complexes. These complexes are generally very stable with low solubilities, making it so that our phosphorus is not actually bioavailable to microorganisms. That highlights why it's really important to understand the interactions of carbonates with our biostimulatory solution and how buffering capacity might help influence the effectiveness of these solutions. The project objective was to understand the role of site-specific properties in the effectiveness of biostimulation in cold region calcareous soils. Specifically, how geochemical parameters condition our biostimulatory solutions, and if there's a way to counteract the effects of that conditioning so that we can get increased effectiveness of our stimulation solutions. The end goal is to create a conceptual model that will integrate site-specific geochemical parameters and microbial activity to estimate hydrocarbon degradation rates. For this study, we collected cores from a site in northern Canada with known diesel impacts. The general setup was the same across all the tests, where once received, the cores were extruded from the plastic casing and cut into 6-centimeter diameter sections. The soil was then inserted into a slotted PVC pipe where we added needle holes on either side of the soil to help infiltration of our biostimulatory solution. Mesh was then placed on both ends and zip tied in place to prevent soil loss. This soil core was then placed into a 120 milliliter glass jar that was filled with solution, either DI water for a control or a biostimulatory solution. Following setup, these microcosms were added to a rotary shaker that was set at 150 strokes per minute in a room with a constant temperature of 5 degrees Celsius, which was used to simulate field temperatures. Then, at specific time intervals, we collected solutions for the analysis of petroleum hydrocarbon content, anions, and cations. At the end of the test, we also did some qPCR tests on the soils to determine microbial composition. Probably the most important part of this test is our biostimulatory solution. We know that we have an added input of carbon from our petroleum hydrocarbons, and that disrupts the soil's natural carbon, nitrogen, phosphorus ratio. So we need to add in nitrogen and phosphorus to balance out that ratio. In this test, we added sodium triphosphate for our additional phosphorus and nitric acid as the additional nitrogen. We also added a couple other important components. The first was magnesium sulfate or potassium sulfate as a terminal electron acceptor. Terminal electron acceptors are really important because they are used by the organisms for respiration via electron transfer chains, and they essentially enable oxidation of our carbon substrates, allowing for microbial growth and subsequent breakdown of our petroleum hydrocarbons. The final component of our biostimulatory solution was ammonium ion citrate. This was added for two reasons. We used the iron as an additional terminal electron acceptor, and then citrate as a low molecular weight organic acid to help chelate our calcium and magnesium ions, ultimately reducing phosphorus mineralization, allowing for increased bioavailability of our phosphorus in solution. Our biostimulatory solutions were optimized based on unique geochemical conditions in our soil, including calcium, magnesium, phosphorus, and pH that were present in those initial soil cores. Using that information, we were actually able to develop a decision tree where we could get down to specific, unique, focused biostimulatory solutions. Now this looks a little bit scary, our decision tree, so I'm actually going to walk through how we got to focus 3, which was used for these 4 cores. The first thing that we did was we looked at soils with buffering capacity. So soils with a high buffering capacity had a pH greater than 4.5 when citrate concentrations were increased from 0.24 to 10 millimolars. In this case, we had a high buffering capacity. The next thing that we looked at was calcium content. Soils with high calcium content had a magnesium to calcium ratio of less than 0.12. This was used to determine whether we needed to use potassium sulfate rather than magnesium sulfate to eliminate any concerns about calcium phosphorus complexation. In this soil, we had a high calcium content. The final item that we looked at was was ferrous iron, looking at if the soil was iron rich or not. An iron rich soil was classified as where we had iron in solution that was greater than 30 milligrams per liter. And if we did have an iron rich soil, we actually eliminated the ammonium ion citrate because it wasn't necessary, and it helped us create a more basic biostimulatory solution. In this case, we had an iron rich soil. And that's how we got from focus 3. We did that for all of the different cores that we used throughout this project. Additionally, we had a number of different controls for each core. We had a DI water control, which was used as a negative control. Then we also used a base solution that had been quite effective at numerous sites in Saskatchewan and Alberta as kind of a positive control. We obviously used our focus control or our optimized solution. And then the final solution that we used on these cores was a slightly oligotrophic solution because we wanted to determine if adding a little bit lower nutrient in our focus solution would be enough to effectively boost our indigenous microflora to start degrading our hydrocarbons. That would allow us to add a little bit less of the acidic inputs so we wouldn't have as much of a change in our pH. For our analysis, we focused on hydrocarbon fractions F2 and F3 because those are the main constituents in diesel. In order to determine the effects of treatment on degradation of F2 and F3, we used a generalized least square model, which accounts for the initial petroleum hydrocarbon concentrations and core depths to allow us to use the residuals to determine degradation rate constants and essentially provide a clearer indication of our treatment effects. This figure here highlights the degradation constant residuals along the y-axis, and then our treatment along the x. We have F2 on top and F3 on bottom. The main item for these box plots to highlight is that in most cases, we see a degradation rate constant above 0, indicating that degradation was actually occurring. We move on to our coefficient estimate, again, F2 on top and F3 on bottom. We have actually a much clearer indication of what is going on. So for F2, we see that we have a statistically significant difference in degradation, or higher degradation rate for our base solution when compared to the other treatments. It's also important to highlight that we do see, while not statistically significant, we do see an increase in degradation in our focus solution above our DI water control and our Focus plus Allego treatment, indicating that we were seeing degradation of F2 in our base and our focus. However, probably the more interesting result is our F3 concentrations, where we see we have statistically significant difference in degradation in our focus and Focus plus Allego treatments, indicating that we had optimized our solutions for site-specific geochemical parameters using our focus solutions. A possible reason for the difference between the F2 and F3 results is that biostimulation works differentially across different petroleum hydrocarbon fractions, and because you have different groups of degrading microorganisms for each of those fractions. So here, we were likely stimulating more of the F3 group with our focus and Focus plus Allego solutions, and F2, we were really targeting our F2 degrading microbes with our base solution. We also tested for nutrients, where we looked at the percent difference from the initial concentration to 28 days and compared it to 0, where a more significant decrease over 28 days meant that we were having organisms actually using our nutrients, where if it was over, then they might not have been using it as much, or we were just providing extra nutrients. So for phosphate, we can see we had a huge variation in our results, but it was kind of all hovering right around 0 or below, indicating that we were probably adding, or that the phosphate we were adding was being used, but we potentially could have added a little bit more to increase biodegradation. It's important to highlight our Focus plus Allego treatment, which was quite low, indicating that we were probably still phosphorus limited in our Focus plus Allego solutions. For nitrate, we actually have a very similar trend, but we actually do see for our base and our focus solutions that that nitrate was actually being used in those solutions. And again, we saw our Focus plus Allego quite a bit lower, and likely we're still nutrient limited in that solution. This trend is actually further highlighted by using the same generalized least square modeling that we did for F2 and F3 for nitrogen, where we looked at nitrogen immobilization along the Y, and again, treatment along the X. Nitrogen immobilization relates back to nitrogen deficits. So what we're seeing is that our nitrogen in our base plus our focus was being used by our microbes. And we were seeing, while not as significant, a little bit of usage in our Focus plus Allego. The next thing that we wanted to do was look at microbial activity and how our biostimulation solutions were stimulating the microbes. We used positron emission tomography for this. I'm not going to go into too much detail because Dr. Schmidt's going to talk a lot more about that in the next presentation. But briefly, when you do a PET scan, you have positrons that are emitted during the decay of a radiotracer, such as 18F. As decay occurs, your positrons travel through the media, such as the soil, losing speed and energy. Ultimately, when enough energy is lost, the positrons combine with electrons, and you have a mass to energy transfer that results in the production of 2 photons that travel in opposite directions. And you have an image that is constructed when those 2 photons hit those different detectors within a defined time limit. For this study, we use an F18 labeled FDG, or fluodeoxyglucose. This was selected because it's used by heterotrophs during the glycolysis pathway. And essentially what you do is you add your radiotracer, you do a little bit of rinsing, and then you're measuring what stays in the system during your PET scan. So essentially you have higher gamma counts, which equates to higher microbial activity. And what we did is we took that radioactivity, which is along the x-axis, and we correlated it to gene expression from our qPCR. This figure really highlights that in our base solution, we were seeing much more radioactivity that correlated to a higher microbial activity. This indicates that in our stimulated system, we're seeing higher activity. and that can likely be related back to higher petroleum hydrocarbon degradation. This is further highlighted by the fact that in our DI water control, we didn't see a change in microbial activity or gene expression when correlated to our radioactivity. So in conclusion, we had general trends that were evident, but there was a lot of different variation in our results, and that's likely due to an interference or dilution effect in our microcosm. So while the trends are a little bit hard to pick apart at times, we do believe that there's further development of those solutions is warranted. And how we can do that is really by evaluating absorption kinetics in our systems. And what we've come up with is a new treatability cell that will actually mimic fractured flow with a fracture aperture of about 1 millimeter, and then also pumping it in at a low volume or low flow rate that will try and mimic hydraulic conductivity in our normal systems. But first, we need to compare the absorption kinetics within our treatability cells to traditional absorption kinetic experiments. We're going to use the thin dismissible experiment to compare the new treatability cells to traditional absorption. The idea behind thin dismissible is that it will track absorption of a molecule over a matrix over time until you hit equilibrium. Further, we really wanted to operationalize the testing of these treatability cells so that we could run more cells with very consistent conditions over a longer period of time. What we've come up with is this low flow distribution system that will actually plump at a rate of about 0.1 milliliters per minute, and then it will actually mix our solution right before it enters into our treatability cells. This here is an image of our treatability cell where you can see that we have our solution flowing on all sides of the soil puck that we're going to be adding so that we can really maximize those surface interactions that we're seeing. The solution will flow around the soil puck and into a collection vessel where we can use it to analyze for cations and anions. And then it will also flow into a second collection vessel where we can collect gas samples for the solid phase microextraction of BTEX as well as other petroleum hydrocarbon fractions. So in summary, the whole purpose of this experiment is to assess biostimulation holistically by looking at site-specific geochemistry as well as microbial activity with the goal to create a model where we can predict an effective solution for all sites, which can only be done by understanding how our geochemical parameters condition our biostimulatory solutions and then building solutions to counteract those effects. A couple of quick acknowledgements. Thank you so much for the statistical analysis of Steve Siciliano and Steve Mamet, as well as the University of Saskatchewan. Soil was collected from a site held by the Site Assessment and Remediation Unit for the Government of Yukon, and Jacobs Engineering completed the fieldwork. And I'll take any questions.
All right, thank you, Amy. So I have a question for you.
Yeah.
Can you talk to me at all about the makeup of your microbial communities? What are you seeing in your soil? I noticed you've done some sequencing. I'm assuming that you have some species identification of your bacteria. So what are you actually finding in there?
That's something that we haven't quite finished analyzing yet. So I'm not 100% sure what the makeup is quite yet. But it's definitely something that's going to be interesting.
I'm guessing that you're going to see quite a bit of Pseudomonas, that sort of thing.
Yeah, more than likely.
Do you know if the makeup of the population is showing changes? Can you tell yet whether you're seeing amplification of a particular type of bacteria or whether it's pretty consistent from start to finish? Can you say anything about that?
Not quite yet. Once we get the gene sequencing back, we'll be able to, and hopefully that'll be part of next year's presentation.
Oh, good. Okay.
Yeah.
Yeah, I'm not seeing any questions in the chat box yet.
Okay.
Yeah. Maybe Amy, talk a little bit about where you are in your program. Are you halfway through?
So I am about probably a third of the way through. We would have been obviously a lot further ahead if the university hadn't closed and kind of remained closed. But this winter will really be amping up this system to get our results. So I'm kind of technically should be farther along, but I'm kind of in the beginning stages, I guess.
Yeah, understandable. Let me see if I can. How did you estimate degradation rates?
That was done through the generalized least score models.
Okay. And I think that's all I'm seeing for questions. All right. Amy, I want to thank you for your talk this morning. Good job.
Thank you.
I'm hoping Michael is waiting on board here. Michael, can you start your webcam so I can see if you're there? I just want to thank TerraStrike again for sponsoring this session. You are in the University of Saskatchewan session, and my name is Liz Murray, and I'm your moderator this morning. So we are waiting for Michael, who's up next. We're going to give him a few minutes. Is it possible for us to show the TerraStrike video again?
Yeah, just real quick, Mike's here. He's just having issues. Oh, he's on, never mind.
Okay. Oh, there he is.
Oh, you guys can see me, all right. Wow,
Yeah,
that sounds pretty aggressive. I'm gonna try to drop in that shade a little bit, I think.
No, you're lucky you got sunshine. Actually, we have a bit of sunshine here in Calgary, but we're waiting for the massive snow to hit tomorrow.
Oh man, you guys are getting snow. I was about to complain
Yeah.
about the weather here, but it's at least we're not having snow yet. So.
All
Okay.
right, we are going to wait a few more minutes here because we're a little bit ahead of schedule. Is it possible for us to show this TerraStrike video again? I'm just waiting to see.
TerraStrike is based in New Hampshire. Our manufacturing facility is outside of Toronto in Brampton, Ontario. And we have done work in 5 nations throughout the world. The majority of our work is in North America, Canada, and the US. Been in 23 states and 4 provinces. We've also been in Japan. We've been in the tannery districts of India. And we have done sludge work in Saudi Arabia.
Great. So thank you to TerraStrike once again for sponsoring the session. Of course, RemTech is made possible by all the sponsors that help out. So I want to thank everybody that's helped out with RemTech. So our next speaker is Michael Schmidt. He's at the University of Saskatchewan. He's a postdoctoral fellow. He's going to be talking to us about positron emitting isotopes for spatial resolution of microbial activity in soils, applications and future directions in in situ bioremediation. So Michael, I'm going to get you to share your screen
Okay.
and make sure that it's on the full screen version.
Does everything look good?
Everything looks good, so I'm going to check out here. If you have a problem, I'll step in, but go ahead.
Okay, great. Well, thank you for the introduction there. So I'm going to be talking a little bit today about positron emitting isotopes. I know Amy kind of touched on this a little bit. I'm going to get a little bit more involved. I'm going to talk about some reasons why we need these imaging approaches, what these imaging approaches actually look like, some examples from our lab group, and also a quick overview of some potential applications moving forward in remediation. First, I'd like to start out with this this rather grim Rembrandt painting. This was a painting Rembrandt made hundreds of years ago, basically showing the way we used to learn about human bodies, the way we used to learn about medical diagnostics and medical treatment. And fortunately, we've come a long way since then, thanks to a lot of these same exact positron imaging approaches that allow us to do non-invasive imaging. And I hope that by the end of this, I can convince you that this is something worth considering moving forward as a mechanism for advancing soil science and specifically bioremediation as well as medicine. So first off, why do we need to be thinking about something like this? As anybody who's worked with soils will tell you, soils are a highly heterogeneous medium. They vary locally, physically in terms of pore structure, which has vast implications for things like water flow and gas transport. And also you get differences in local biogeochemistry. You get different speciation across soil samples, different elemental distribution, things of that nature. And you also end up with different distribution of microbial communities in soils. And this is really important because some of these microenvironments, be it driven physically or biogeochemically, tend to favor certain microbial activities over another. And within the context of bioremediation, we can consider things like contaminant transformation. Unfortunately, though, when we look at the bulk measurement of these properties and these activities with respect to soils themselves, they oftentimes don't correlate particularly well. And even if they do correlate well, it's still the underlying spatial relationship between the microbial activity and the localized microenvironment in which it's favored is absolutely lost. And unraveling these spatial relationships in the long term is going to be critical to understanding the true processes that underlie the activity we see. And I believe, and I'm going to present some examples to support this, that positron imaging may help reconcile these points. First, what is positron imaging? What is this I'm talking about? And I think before we can really address that, we have to touch on the nature of the positron itself. If you pick up a dictionary, this is the definition you're going to get for positron. Positron is a subatomic particle with the same mass as an electron and a numerically equal but opposite charge. So in essence, it's the antimatter of the electron. So it's a positively charged electron. You can think of it that way. And they're formed by the decomposition of what is known as a positron emitting radioisotope. And an example of this is shown right here, where we have carbon, and we have carbon-11, which undergoes a shift of one of its protons to a neutron. It becomes boron-11, and in the process yields an electron antineutrino, which we're not particularly interested in here, and the positron. And isotopes like this, like carbon-11 or nitrogen-13, oxygen-15, or the also commonly applied fluorine-18, are quite unique. They're unique in the sense that they are not naturally occurring. They're probably a little bit unusual if you've never heard of them before. And they need to be produced at cyclotron facilities, of which there are 20-something spread across Canada. So these isotopes are available to use for research experiments. And they're made through the acceleration and bombardment of matter. One of the unique things about these isotopes, though, is that they have very short half-lives. I mean, if you look here at like oxygen-15, for example, it has a half-life of about 2 minutes, which has both benefits and drawbacks experimentally. Great, so now we have a general idea about what the positron is and how we make positron emitting isotopes, how do we actually visualize them in samples? And this figure here is going to look fairly familiar, because Amy showed something quite similar. And basically what it's describing are the two primary strategies we use for imaging positrons: positron emission tomography, or PET, or autoradiography, which is the other approach. And PET, as Amy more or less touched on earlier, takes advantage of the physics of the positron in materials. So what happens is we have our radio tracer, our radio positron emitting isotope, undergoes decay, releases the positron, and the positron will travel a certain distance in the medium in which the tracer is present until it loses a sufficient amount of energy, at which point it will undergo what is called annihilation with an electron, in which both the positron and the electron are destroyed, and an X-ray is given off, well, two X-rays, excuse me, are given off, two gamma rays are given off in an anti-parallel orientation from the site of annihilation. And if we have an array of gamma detectors, we can actually get a three-dimensional tracer distribution within a sample. And an example of an imaging system that we've used in the past is this PhytoPet system that our collaborators from the University of Regina have made. It's a pretty cool system. We see a collection of 4 detectors that rotate, and they move up and down, so we can get a really nice three-dimensional distribution with that. The other imaging modality that's commonly used to image positrons is known as autoradiography. And instead of PET, which images the gamma rays formed from annihilation, autoradiography directly images the positron itself, which has an advantage of better spatial resolution, because we're not waiting for a travel to occur before annihilation. But it also has the disadvantage of only being two-dimensional because of the nature of the exposure. So principally, autoradiography is very similar to developing film, where you take a radioactive sample, put it in contact with a film, and allow the radio tracers to kind of develop on the surface. And an example of this is this, yet again, a very grim image of a cross-section of a rat brain that's been dosed with I believe, radioactive sugar, and we can see the tracer accumulation in that rat brain. So what are the overall benefits of these positron imaging strategies? Well, one of the big things is that the radiation that's formed penetrates well in many media, which is nice. Because if we're considering something like soil, which is usually opaque and we can't actually see it visibly with visible light, we can actually visualize tracer movement inside these, which is pretty nice. On a few slides ago, I showed a table with, I think, 4 or 5 different isotopes, but there are many, many more available. And these isotopes themselves can be attached to molecules, or they can be bound to chelators or something. It really opens up a lot of chemistries, which allows us to kind of do more tailored tracer applications to a variety of different systems. And as I alluded to previously, these isotopes are strictly synthetic. So what we see in a system is only what was added. There's really not much of a natural background for these. And by not much of, I mean there's basically none. And the short half-life of these radio tracers is kind of a double-edged sword. One of the benefits of it is that they have such a short half-life, so if these tracers are being retained, it's usually by some form of an active process. One of the things that I find particularly cool about this as a chemist is we're able to detect these tracers at really, really, really low concentrations. So we're looking at like sub-picomolar type concentrations, which is really nice for environmental systems where we're perhaps dealing with a trace contaminant or something of that sort. And we can apply a tracer with minimal disturbance to sample chemistry and in a more environmentally relevant concentration. And lastly, you can do repeated scanning or imaging, and this is particularly so with PET, but you can also do repeated exposures with autoradiography, and you can kind of develop some kind of a concept of the time resolved distribution of these tracers. So you can see perhaps tracer movement through time in a sample, which is kind of nice. And because of these benefits, positron imaging, it has been widely applied in the past to things like medical diagnostics. We can see here there's a patient who again was treated with a radioactive sugar, and we can see accumulation in tumors in this individual. It's been really useful in plant science, where we have some two-dimensional radiographic images here of radioactive carbon dioxide uptake in a variety of different leaves. And also veterinary medicine, where I like to joke this is a PET scan. It's a PET scan of a golden retriever that has also been treated with the radioactive sugar. And we can see that there are some locations of accumulation in this specimen. So we kind of now know the basics of the technique. We know some of the benefits of it. So let's move on now to some examples from our lab to show how it can be applied more directly to soil science and ultimately bioremediation. So one of the big questions that soil scientists oftentimes are interested in when it comes to soils is what is the activity of the soil? What is the kind of net metabolism of the soil? And one of the ways we like to get at this is using what's called a carbon dioxide evolution assay. And principally what we do is we just take some soil, we kind of put a bit of water on it, it's basically the field capacity. We put it inside a bottle and we see how much carbon dioxide is evolved through microbial respiration. But if we consider this situation here with this this nice soil sample we have here in this bottle, we can probably get more out of that. I mean, if we can say, for example, 1,000 micrograms of CO2 have been evolved by the soil sample, where is that occurring within the soil sample? Is it spread fairly evenly throughout, or is it localized kind of like in the center example closer to this plant root, or is that strange dark hole in the middle of the soil sample where all the carbon dioxide is coming out of? And unfortunately, with existing techniques, a lot of these local soil environments aren't resolved. And again, yeah, most of the methods for measuring these are either destructive or they really just don't have that spatial component. So to kind of address this fairly basic question of mapping microbial metabolism, we applied a positron emission tomography to this question. And in this specific example, what we did is we took two sand columns, one of which was inoculated with active Pseudomonas fluorescens, and the other one, as a control, was inoculated with an autoclaved sample of Pseudomonas fluorescens. And what we did is we passed growth medium, basically a basal growth medium, through these columns, and we dosed it with a radioactive sugar. It's 18F labeled glucose, basically, and the microbes will eat it up as they would regular glucose. Although the metabolic pathway isn't entirely clear, they are known to metabolize this sugar. And what we did in classic radio tracer style, we dosed our system, we pulsed our tracer in, then we chased it out. We rinsed with growth medium again, such that the only FDG that was retained in the column was retained by active bacteria. And then after this point, the sand cores were imaged with positron emission tomography using that detector array that I showed you earlier. And we were actually able to visualize some of the activity in that core. And we can see that the core with the live Pseudomonas has a lot more of retained activity than the autoclaved control. And furthermore, we can see that in three-dimensional space, we can actually resolve those regions of activity fairly well. So yeah, this is a fairly basic idea of the kind of things we can learn with positron imaging. So we showed an example right there of use of positron emission tomography to identify where the activity is. That can only take us so far. And again, if you remember back to the first slide here, one of our first goals is to kind of reconcile the regions of activity that we see with bulk properties. So we need to be able to kind of measure what's going on in these specific areas. And some things that have previously been done in the literature are a couple different things that kind of allow us to further interrogate those regions. A really nice example of this on the left is this is a 3D image of plant roots, corn roots of corn plants that have been treated with 11CO2. And you can see that the activity has been transferred to the roots. And once the active roots were identified, the authors took the root and they extracted sugars from it to get a profile of where that recently fixed carbon ended up in terms of the sugar profile. In the center here is an example of targeted genetic analysis as guided by positron emission tomography. And this was an experiment fairly similar to the one we just showed, where a microbial active sediment, so an active sediment, I believe from a river bottom, was taken and incubated with radioactive sugar, again 18F-FDG. And what they found in these active regions was some changes, some shifts in the microbial community. So they did a targeted community analysis there, which I think is quite nice. And then lastly, and kind of going back to the first slide where we talked about local biogeochemical factors that could be driving activity. You know, we could, once we identify a region with PET or autoradiography, we can kind of take a closer look with some focused chemical and physical imaging, such as X-ray fluorescence, which will give us a better idea of the elemental distribution in these soil samples, or also microdiffraction if there is some kind of a mineralogical driver of the process. So these are some examples from the literature here of basically the kinds of things we might be able to do. But we do also have some examples from our lab as well. And one example of this is the experiment that Amy just actually described quite well. So I hope I don't overlap with what she said too much. Where we had some soil samples from up north. I believe they were diesel contaminated soils, and they were the cores were sectioned and they were exposed to either a variety of biostimulatory solutions or deionized water. And after incubating for a while, these cores were treated with 18F-FDG again to kind of allow us to visualize heterotrophic activity in these soils. And what we saw is the soil cores with the biostimulatory solution had altered microbial distribution. That's what PET showed us. But when we took those cores and we sectioned them up, as shown here in this image, these pictures here across the bottom, and then we did a subsequent genomic analysis based on the activity, we can see that the activity distribution that lined up with the PET images was backed up by genetic analysis, wherein we saw this beautiful correlation between the radioactivity as shown by PET and the 16S rRNA that was able to be extracted and expressed in those soil samples. But again, to kind of bring it back out to our reasons for doing this in the first place, you know, we're able to kind of make some really important observations with respect to some of the bulk measurements as well. And we saw that the bulk measurements, such as microbial activity, they increased with treatment with some of the biostimulatory solutions. Nitrogen uptake and immobilization, which again is another way of kind of saying, well, the microbial activity is potentially going up. And we saw for some of the stimulatory solutions an increase in hydrocarbon degradation rates. And I think right now, Steve Mamet, one of the research associates in our lab, is in the process of kind of reconciling these bulk measurements with some of these PET measurements to put together a manuscript. So I'd be very interested to see what they come up with. This is another example from our lab where instead of doing targeted genetic analysis, we do some more targeted biogeochemical analysis instead. And this isn't quite as directly related to bioremediation, but I think it kind of, it's a framework that'll apply quite well to it, I think, in the long term. So we have some soils from the high Arctic up I think off Ellesmere Island, I believe, some Arctic soils where we took the soils and we were trying to figure out the distribution of autotrophic and methanotrophic bacteria in these soils. So I guess for our general dosing strategy, we took these soils, kept them at room temperature and about field capacity moisture. And we incubated them for a couple days to get them nice and active first. And then we exposed the samples to a radioactive tracer, which was basically ambient air with either picomolar levels of 11 CO2 or 11 C methane. Then we flushed the system clean and we exposed the soil samples to radiographic imaging film. And some examples of the raw images are presented here, where we can see discrete regions of activity for both carbon dioxide and methane uptake in these soils. And once these images were kind of processed and reclassified, we were able to identify hotspots where the methanotrophy and autotrophy were specifically occurring themselves. And what we did is we isolated soil samples from these cores, kind of in correspondence with these images, and did targeted biogeochemical analysis at the Canadian Light Source synchrotron up here in Saskatoon. And more specifically, we did local carbon and nitrogen speciation to kind of get some idea of the organic matter speciation, or in the case of carbon, also some carbonates. And for nitrogen as well, some idea of potential nutrient-related drivers. And we also did iron X-ray absorption spectroscopy to help characterize some of the iron mineralogy, to help kind of address this hypothesis that there could be a mineralogical driver to the distribution of these communities. And principally, we kind of discovered two different things here. One was for where the sites of methanotrophy. Now, relative to the regions where there was no activity, regions with methanotrophic activity had a greater proportion of inorganic nitrogen present, which suggests some kind of a nutrient control on the distribution of this activity in soils. This is a little bit different from the autotrophy or the locations of carbon dioxide uptake, where we saw that relative to the background where there was no radio tracer taken up, the regions of carbon dioxide uptake were associated with greater presence of carbonate minerals, as well as shifts in the iron mineralogy, suggesting more of a mineralogical control for the autotrophs in these Arctic soils. So I'll start kind of moving into the summary portion of this presentation now. And kind of when we're thinking about positron imaging and what it can tell us about soil science research, I think it's important to kind of step back and think to some very elementary lessons. And in this case, I mean that very literally. I know back when I was in elementary school and we were taught to start thinking about questions, we kind of start with the 5 W's, and the 5 W's, according to Wikipedia, are questions whose answers are considered basic in information gathering or problem solving. And Wikipedia specifically identifies research as an important area where the 5 W's apply. So it's legit. If Wikipedia says it, it's legit. And the 5 W's, in the event that you do not remember, are who, what, when, where, and why. And I believe that positron imaging is really a great tool to help address all these W's. So if we can apply the appropriate radio tracer chemistry, we can get the what is happening. We can really key into a specific process of interest with the right radio tracer chemistry. And if we get spatial visualization with some kind of degree of time-resolved characteristics, we can say not only where did it happen, but we can say when it happened. And then if we follow it up with some kind of a targeted genomic analysis, we can say who is there. So who's making this happen from a microbiological standpoint. And ultimately, if we're trying to figure out who is there, we want to know why they're there. And again, if we're looking at possible biogeochemical factors, I think we've again demonstrated a framework in which we can identify those as well. So moving forward with bioremediation, I'm going to show a couple of, I'm going to describe really quickly a couple of examples that we're working on in our lab, and also another potential example where I see some possibilities here. And to kind of reference the previous slide, I think a lot of bioremediation, a lot of those questions are going to be addressed basically by modifying the what. And what I'm going to present here is going to be based largely on what I know we can do in terms of tracer chemistry, and I believe that there really are a lot more possibilities out there. And one example that I know there's a PhD student in our lab kind of getting his feet wet on this is remediation of BTEX compounds using 11 carbon labeled toluene, maybe 11C benzene as well, with the intent of imaging BTEX degrading bacterial communities in soils. So getting a more targeted, rather than relying on a general heterotrophic activity, really keying in on that with specific contaminants. And something I've been kind of working on developing with some of the students and technicians in our lab is utilizing 11 CO2 tracers and 13 nitrogen tracers to image autotrophic ammonium oxidizing bacteria activity in ammonium contaminated prairie soils to help us understand the biogeochemistry, and more targeted isolation of communities in some of these prairie soils from Western Canada. And lastly, this isn't something we've really touched on so much yet, but it's an area that I see a lot of potential for, as there's been some evidence that suggests that methane oxidizers have the capacity to reduce or oxidize chlorinated organics depending on their local environment. And I think we feel pretty confident we have the ability to make a really nice methane 11C tracer at the moment, which could be used to help perhaps visualize this process as well. So I think to kind of bring it back to the intro slide here, when we kind of, as soil scientists or remediation scientists, inevitably find ourselves in the role of Rembrandt subjects, of kind of picking apart and dissecting and learning about soil samples, hopefully we can kind of start to be a little bit more informed about it when we start going in and actually playing the role and doing the dissection and doing the destructive work, and understanding what's going on inside soils, like the living organism more or less is. So on that note, I would like to quickly acknowledge the two lab groups I work with here at the University of Saskatchewan, the labs of Derek Peak and Steve Siciliano. In particular, Curtis Sanger is a technician in Steve's lab who has done a lot on the radiochemistry side of things. Dr. Catherine Stewart as one of our collaborators at the U of S. And these guys here at the University of Regina, Aram and Mayran, these guys are absolute physicist wizards. They can do all the electronics and imaging stuff, and it's really quite impressive, and really owe a lot to them. And we're fortunate enough here to have a cyclotron facility, so a place on campus here where we actually can make these isotopes. And in particular, at that facility, Dr. Jackie Cawthray has been really helpful. And I've gotten funding through the Fedoric Center and NSERC, and also the Federated Co-op as well. So at this point, I hope there's some time for some questions. That's it.
All right. Thank you, Michael, for that.
Sure.
So I have a couple of questions. The first is there anything in the soil that interferes with the ability to detect the tracers? Do you get in or get out what you put in, or are you finding that there's a particular contaminant or another type of problem that might interfere with being able to detect those tracers?
Yeah, that's a really good question. So one of the things that is a major problem, especially if we're using positron emission tomography in soils, is the gamma rays that are formed, they can become attenuated by either scattering or absorption processes or something by the high density medium of the soil itself. So oftentimes the radiation can actually have a hard time getting out if you're using a really dense soil.
Okay, and to follow on that, I get that half-life is probably one of the determining factors on what isotope you're going to use, but other than half-life, is there some that work better than others for doing this kind of work, or is it mostly dependent on what you're looking for, like a CO2 versus something else kind of thing?
Yeah, so half-life is a factor, but we also have to consider the similarity of the labeled molecule to what we're actually trying to study itself. So for example, we could take something like benzene, for example, and tag it with an 18F, or we can try to find a way to incorporate 11C into the benzene structure itself. So we know that we actually have what is chemically benzene, instead of having it fluorine labeled benzene. So we can have a little more confidence in the behavior at that point. But one of the drawbacks there, as you kind of alluded to, is we have to kind of struggle then with the half-life, whereas carbon has a 20-minute half-life, and F18 you can get a 2-hour half-life out of. And realistically, for these experiments, we can expect, I don't know, usually as far as like really good solid imaging, maybe we can work with these things for 10 half-lives or so. So for something like oxygen, that looks like 20 minutes, and for something like F18, it looks like about a day. So it really kind of, yeah, you do have to kind of put that into account when you're designing experiments. So yeah.
Okay, I have a question here. What are the special considerations in collecting the soil samples for using this method?
Oh, that's a good question. I mean, I think it depends what you're trying to ask. So, if you're trying to use like a liquid type tracer, like a 18F FDG, it's really good to have a sample where, you're not dealing with like a hard, compact clay, for example, where water movement can become really restrictive because your tracer won't be able to get in. So sometimes it's really good if you can get a core and keep it intact. That oftentimes works really well rather than trying to backfill, because oftentimes if you take like a crushed soil sample and try to fill a core with it, you're not going to have a lot of those natural preferential flow pathways and things that will allow really good permeation of the radio tracer into the sample. Now, it's a little bit of a different situation though if we're using a gaseous species like 11CO2 or 11-methane, where gases obviously diffuse and move through soils a lot more quickly and pervasively. So.
All right. Thank you, Michael, for that talk. And
Sure.
Yeah, I think our next speaker is Aimee Schreier. She's also from the University of Saskatchewan. Just going to wait here until we get her queued up. Yeah, and I'll get Aimee to share her webcam. Welcome, Aimee. So we are going to listen to a talk about electrical conductivity in soil as an indicator of biostimulatory solution dispersal and pHC biodegradation potential. So I'm going to turn it over to you and get you to share your screen, and I'll step in if you have any problems.
Okay. You can see the presentation then? I'll assume yes.
Yep, we are good to go.
All right. I can see why it's confusing because, yes, all this is very new. So I'm just going to go ahead and get started on this, where we're looking at electrical conductivity in a greater scheme of what we call the stall investigation. So I'll get started on that. So biostimulatory solutions are used to promote or prompt microbial communities to degrade petroleum hydrocarbons. In theory, with the addition of nutrients and sufficient time, the microbial communities are unrestricted by nutritional limitations and can consume the contaminants from the system. However, what is often the case is that some areas of the site will be responding really well to the nutrients, while others won't be doing anything of interest within the desired time. This means that that specific area is designated as stalled. So these sites are really often dismissed as unsuitable for biostimulation and other means of remediation or pursuit. Once that decision has been made, there is often no other further investigations as to what caused the decreased degradation of petroleum hydrocarbons. So we are interested in examining the soil further to understand its remedial potential. This includes determining whether there's any positive indicators of remediation processes, or could there be indicators such as early warnings that may tell us that a site's going to be stalled or is already stalled right now. So the first thing we needed to do was understand the petroleum hydrocarbons conditions on our sites which are undergoing remedial processes. So we have compiled a large database that you see here since 2015 for sites in Saskatchewan that are undergoing biostimulatory treatment. There's a total of 930 soil samples in what we affectionately call the stall database. So in the future, when I call something the pre-amendment period, it refers to the time where we didn't start remedial treatment, which could vary by site from 2015 or 2016. But the important thing is to know that this is before we started treatment. Each sample, we're going to be using terms like location. And what location refers to is a specific area that's been sampled at each time point during environmental assessment, and areas where they were sampled one year but not another were excluded from this soil stall database. So another way to think about it is boreholes. You know, we usually watch the same areas and give them borehole names, but those tend to change over time and they got really confusing and lost track of the actual names. So we call them locations instead. We use the incremental sampling methodology to measure the F1 BTEX concentration. And I'll go into a little bit what is ISM, and I'm going to call it ISM from now on. So incremental sampling methodology is an improvement over discrete sampling, as discrete sampling is incapable of accurately representing site conditions. So if you see the figure on the right, if we pretend the box is a core and we're taking two discrete samples from the random area, those samples would have poor spatial coverage. It would not address the soil's heterogeneity and would not address the spatial variability of the contaminants. Therefore, ISM is an improvement because we obtain a single sample for analysis that represents the broader area of our sampling core. So this new figure that we see on the right is how we would sample a core using ISM. We take a multitude of increments, combine them together, and obtain that single sample for further analysis. This looks like composite sampling, and ISM is a type of composite sampling, but it's not the same. ISM is an improvement over composite sampling because you need to have equal volumes of soil from each area, and you need to make sure you measure and take account into how much soil you're actually putting into that single sample. So ISM reduces the fundamental error associated with our Western Canadian soils. All right, so I think everyone here knows just how difficult environmental data can be. So if we look at this one location over here on the left-hand side, based on drilling and however it went, we had different numbers of cores, sample aliquots at one single borehole. That's fine. We look on the figure on the right, we see that this data is heavily left-centered and skewed and very zero inflated. This is, again, very typical of environmental data. If we can't transform the data as log normal, and obviously it's not normal right now, we can't get rid of points as outliers because then we would be losing information about potential hotspots within the soil. Another problem we see with environmental data is non-detects. So with analytical instruments, it's often pretty arbitrary what's the lower limit of quantification. So if we see the figure on the right bottom, the laboratory detection limit is 0.2 ppm. But if it falls below 0.2 ppm, it's not registered. Does that mean it's a zero? Absolutely not. And this is especially important as a lot of our regulatory standards are below what most analytical instruments can handle. So we call those non-detects. To overcome this issue, we use the hurdle model, and we used it to determine the mean representative of site conditions for benzene. So my lab has published a paper regarding the hurdle model. It's right here. You can get script through this paper as well if you're interested, but I'm going to give a short little rundown of what the hurdle model does. So it splits that data into two groups. One group is with non-detects, so the stuff below the lower limit of quantification for the instruments, and determines based on the spatial dependency of the site, what's the true value of that non-detect. So if you were to take a sample from an area near a tank nest with previous environmental contamination, it's coming up as 0, but everything around it is contaminated, chances are it's not a true 0. it's probably going to have a bit of contamination in that area. If you're to take a sample 6 meters away, 6 kilometers away actually from the tank nest in an area that's never been impacted, it's grasslands, then chances are that 0 is a true 0. So the model takes into account that and creates new values based on that information that we give it. And then the rest of the information is transformed using the gamma distribution. From there, we receive new means and confidence intervals and can determine at each location whether a site is remediated, or stalled. So following data transformation, we created averages by soil zones. So the vadose zone, capillary zone, unsaturated zone. This will provide more insight into the site's conditions and the dependency of such things like soil water and distance from the mineral surface. So I just wanted to give you a little bit more background into what we considered stalled versus remediating or remediated. So as you can see, this is from the same site at 2 different locations, with the figure on the left showing a stalled condition and the figure on the right demonstrating a remediated condition. We have to take into account the distribution of the benzene concentration from the pre-amendment period, which is 2016 in this case, up until 2019. So if there's any overlap in the distribution from the hurdle model, then we would say that the site is stalled. So on the figure on the left, there is distribution, or there is overlap of the distribution for each year, meaning it is a stalled location. The red line just shows the regulatory standard for Saskatchewan. On the right-hand side, there's a high concentration of benzene in 2016, but it's come up to below regulatory standards. In the following years, there's no overlap in distribution. We would consider that remediated. So going back to my 4 sites, I have categorized each zone based on that overlap or no overlap of the distribution and the presence of or absence of F1B techs over the 4 years. So if a zone's been labeled as always clean, no contaminants were ever measured. If it's been categorized as remediated, there was contamination in the pre-amendment period, but it's since been measured as below regulatory standards from based on our 2019 data at the very latest. If it's remediating, it's outside the distribution zone of our pre-amendment period, but it's not 0 yet. Now we look at more stalled conditions. So if it's remediating and now stalled, we saw the distribution of contaminants outside of the pre-amendment period for later years in either 2017 or 2018, but nothing much has changed for our 2019 year, which is our most current year. Now stalled, we would consider that from the pre-amendment year, there's been only overlap of further concentrations of contaminants from pre-amendment year all the way to current. So if we were to compare 2 sites, we can see here that in site 1, there are areas that are remediating. The biostimulatory solution is working in some spots. However, about half of the locations in the site might need further tailoring, as it worked in the beginning, but now we're not seeing any movement of contaminant concentrations, or it might need something completely different because we have always had stalled conditions on site. If we look at our most difficult site, we can see that it's always been stalled. We might need a new approach to biostimulation, as those concentrations have been pretty steady over the last 4 years. So there has been degradation at our sites during biostimulatory treatment over time, but it is very variable, which is the story for a lot of sites. The cause of the variability is not yet known. So if we think about what we're putting into the sites, it's a bunch of salts. It's nitrogen, it's phosphorus, it's magnesium, whatever the site needs in order to make up that nutritional requirement for microbes. Therefore, a simple way where we could potentially use as a proxy for microbial community activity is with electrical conductivity. So in short, electrical conductivity is a measure of the salt quantity in our soils. If we have a low concentration of electrical conductivity, there's not a whole lot of water extractable salts within the system. On the flip side, if we're measuring a high electrical conductivity, there's a greater concentration of water extractable salts in the system. So if we take that information and apply it to our sites, if the biostimulatory delivery system is the black circle on the left, we would assume that the greatest concentration of salts would be near the system. That greater salt concentration means more nutrients are available for microbial communities, and there's no limitations now for optimal degradation rates. If we go larger distances away from the biostimulatory delivery system, we get more issues such as preferential pathway makes it so areas aren't in touch with the biostimulatory solution, or processes like fixation for key nutrients like phosphorus becomes more likely as you get further away. Therefore, what we want from the farther away issues, we would assume that the nutritional imbalance caused by petroleum hydrocarbons have not been addressed, and so forth we would have suboptimal degradation rates. Based on these assumptions, can we say that electrical conductivity be used as an indicator of biodegradation? So early results, I'm going to share a couple of them here. So Site 2 has been degrading very well. If the top two figures there are from the pre-amendment period, and the bottom two figures are our current most recent data, we can see differences in benzene concentration, which is on the left, those two figures on the left, and salinity with the figures on the right. Keep in note the scale. The scale just refers to how much I had to change the size of those blue balls so they would actually fit on the screen. But they're just used so you can understand how the mean of that parameter has changed over the years. The boxes, that coral box represents the former tank nest, and the red circles are the north and south infiltrators. So there is degradation of benzene at site number 2, and it's going very well based on the average benzene concentration, and salinity might be a good indicator of that activity, as near the infiltrators there's been an increase of total salinity over time. When we look at site 4, this one was, if you recall from that table, the one that's been stalled or remediating now stalled. So you can see there is degradation of benzene, where the box there represents the infiltrator net. No, sorry, the drive point network. And the box and those little, the triangle and the diamond in there are just structures on site. So degradation of the average benzene concentration is definitely not as rapid as the previous site that I showed. It's still happening, and here we can say that it's harder to tell how electrical conductivity can refer to benzene degradation, especially that we see that it's quite variable here and a little bit more difficult to understand what's going on. So the jury is still out on whether electrical conductivity will be a suitable indicator for petroleum hydrocarbon degradation efficiency. However, I believe it looks promising. So you remember how many samples I have? It's quite a bit. And having a large database then lends itself to a whole bunch of different avenues that we can investigate from these soils. So we're going to look at parameters with ground soil, which is mostly what we're doing right now during limited lab services, and fresh soils in the future. And one thing to consider is, could there be a combination of any of these parameters with electrical conductivity that would indicate degradation? So at the end of this investigation, we hope to find specific variables that will tell us whether soils will respond either positively or negatively to biostimulatory remediation attempts. As we know that one size doesn't fit all, another way we could monitor it is to see if a solution was working in the beginning, is now not working partway through. Can we check it? Can we adapt and just ensure that we are getting those key microbial activities continually working and driving in the way that we want? So at this time, I will take questions. That's my puppy. He's 10 months old.
All right, thanks for your talk, Amy. So just so all you Saskatchewan fans didn't feel left out, Gainers. All right, I have some questions for you.
Okay.
So the first question I have is about stalled degradation. Has any work been done by perhaps adding plants to those areas to see if that can stimulate degradation again? Do you know if vegetation or root systems to support the bacteria would help with degradation?
A lot of the impacts are pretty far underground. I don't think we've looked too much at phytoremediation right now. We have some pretty nice biostim systems on site in order to promote microbial activities, but right now they're basically all either near operating facilities, which would probably be kind of hard to start any sort of above ground work within the system.
Okay, the other question I have is, we're talking about high EC potentially leading to optimal degradation rates. Can you say anything about what the limit of the EC would be?
What the limit? So
yeah, how high can you go?
Oh, obviously, if we go too high, we're gonna stall site conditions. Because too much salt is fairly straightforward, negatively affects microbial communities. The biostim solutions that we do pump in the site are fairly low saline concentrations, and the most part, my concentrations that I measured average around like 1.3 milli, S per centimeters. So it's never been a really high concentration as of yet over the 4 years. So it is something to be mindful of, not to just dump a whole bunch of nutrients into one area at once. But it's not been a problem yet, but we are mindful of potential toxicity related to salt concentrations.
Okay, another question is the amendments that were added at the 4 sites, were those just nutrient amendments or did you also put in additional microbes?
They were just nutrients. No microbes were pumped into the system.
Okay, have you given any thought to adding microbes, or is that just not on the table kind of thing?
I don't think we've been looking at adding microbes right now. We haven't talked about it. I think we just want to promote whatever's living in the system, and instead of further complicating the site. But it's something.
Great. I don't see any other questions have come up yet. So,
Okay.
yeah. So tell me about your dog.
He is a large Munsterlander, and those are upland hunting dogs. So he's, well, my dad's dog. But I have stolen him for cuddles and walks on many occasions. And yeah, yesterday he found a coyote pup, so and played with the coyote, not knowing it's a coyote. Probably was wondering why this little puppy wasn't interested in playing with him.
Too funny. All right, so I don't see any other questions here, so we are going to wrap up the session. I again want to thank TerraStrike for sponsoring this session. I think we are scheduled for a 10-minute break here, and hopefully everybody will be back to join another session at 10 after 10. Thank you to the speakers today, and thank you everybody for attending.